Good evening, class. Welcome back to Microbiology. This is Silvana O'Coney, and in today's lecture we're going to take a look at prokaryotic reproduction and growth. First, we'll focus on binary fission, which is the type of cell division most frequently observed among the prokaryotes. Cell reproduction leads to an increase in population size, so we're going to consider population growth. Then we'll take a look at the influence of environmental factors on microbial growth. Now let's move on to the effects of temperature. And as you can imagine, temperature has an enormous effect on microorganisms, just like all other organisms. Actually, microorganisms are particularly susceptible because their temperature varies with that of the external environment. And one of the most important factors influencing the effect of temperature on growth is the temperature sensitivity of enzyme catalyzed reactions. You probably remember that every enzyme has a temperature at which it functions optimally. At some temperature below the optimum, then the enzyme ceases to be catalytic. As the temperature rises from this low temperature, the rate of catalysis starts to increase. And when all enzymes in a microbe are considered together, as the rate of each reaction increases, metabolism as a whole becomes more active and the microorganism grows faster. However, beyond a certain point, further increases in temperature actually slow growth, and then sufficiently high temperatures are lethal to the microorganism. So what are the effects of high temperature? High temperatures damage microorganisms by denaturing enzymes and also other proteins such as transport proteins and so on. Temperature also has an effect on microbial membranes, biological membranes. At very low temperatures, biological membranes such as the plasma membrane start to solidify. At high temperatures, the lipid bilayer of the membrane simply melts and disintegrates. So in summary, when organisms are above their optimum temperature, both function and cell structure are affected. If temperatures are very low, function is affected, but not necessarily structure or cell chemical composition. So because of these opposing temperature influences, microbial growth has a fairly characteristic temperature dependence with distinct cardinal growth temperatures as shown on this slide. That is a minimum, optimum and maximum growth temperature. And although the shape of the temperature dependence curve can vary, the temperature optimum is always closer to the maximum than it is to the minimum. Microorganisms can be placed in one of 5 different classes based on their temperature ranges for growth, as shown here. So the first group, the psychrophiles, these are microorganisms readily isolated from Arctic and Antarctic habitats. The next group, psychotrophs. This group includes both bacteria and fungi that are major factors in the spoilage of refrigerated foods. The third group, mesophiles. Most microorganisms are in this category, and almost all human pathogens are found in this group. Thermophiles are found in compost heaps, heating haystacks, and hot springs. And the final group, hyperthermophiles, these are found in and around deep sea thermal vents. There's some evidence that these microorganisms can grow and reproduce at 121 degrees Celsius and survive temperatures of 130 degrees Celsius for up to 2 hours. So let's look at some of the mechanisms employed by thermophiles that enable them to survive in these super hot environments compared to mesophiles. Thermophiles tend to have more heat-stable enzymes and protein synthesis systems, which tend to function properly at high temperatures. And these proteins are stable for a variety of different reasons. Heat-stable proteins have more highly organised hydrophobic interiors, they have more hydrogen bonds, and also other non-covalent bonds strengthen the structure of the protein. Larger quantities of amino acids, such as proline, also make the polypeptide chain less flexible and thereby strengthening it. In addition, the proteins are stabilised and aided in folding by special proteins called chaperone proteins. There's also some evidence that in thermophilic bacteria, the DNA is stabilised by special histone-like proteins. Thermophile membrane lipids are also quite temperature stable. They tend to be more saturated, more branched, and of higher molecular weight. And this results in an increase in the melting point of the membrane lipids. You might recall that archaeal thermophiles tend to have membrane lipids with ether linkages, and this protects the lipids from hydrolysis at high temperatures. And you might also recall that sometimes archaeal lipids actually span the membrane to form a rigid, stable monolayer rather than the typical bilayer. In this table, you can see how the cardinal growth temperatures vary considerably between different groups of microorganisms. We have some examples here of non-photosynthetic prokaryotes, photosynthetic bacteria, protists, and fungi. Now let's move on to our next environmental factor, oxygen concentration. And before we go on, I want you to consider one important question. Why is it that oxygen is so critical for some organisms and it's toxic or even lethal to other organisms? This is the question we're going to try to answer here. So the importance of oxygen to the growth and viability of an organism correlates with that organism's metabolism, in particular with the processes it uses to conserve energy supplied by its energy source. And almost all energy conserving processes in living organisms involves the movement of electrons through an electron transport system. And the nature of the terminal electron acceptor is related to the organism's oxygen requirement. So let's look at the different classes defined by an organism's requirement or otherwise for oxygen. So to the left we have the classification of obligate aerobe. An organism that's able to grow in the presence of atmospheric oxygen is an aerobe, whereas one that can grow in its absence is an anaerobe. Almost all multicellular organisms are completely dependent on atmospheric oxygen for growth. That is, they are obligate or strict aerobes. You know that oxygen serves as the terminal electron acceptor for the electron transport chain in aerobic respiration. Our next classification, facultative anaerobe. Facultative anaerobes don't actually require oxygen for growth, but they grow better in its presence. In the presence of oxygen, they will use aerobic respiration. Our third classification is the aerotolerant anaerobe. Aerotolerant anaerobes such as Enterococcus faecalis simply ignore oxygen, but they grow equally well whether it's present or not. In contrast, microorganisms belonging to the next group, the strict anaerobes, they don't tolerate oxygen at all, and they actually die in its presence. Aerotolerant and strict anaerobes cannot generate energy through aerobic respiration, and they must employ fermentation or anaerobic respiration for this purpose. Finally, there are aerobes called microaerophiles, and these microorganisms are damaged by the normal atmospheric levels of oxygen, which is about 20%, and they require oxygen levels below the range of, say, 2 to 10% for growth. The nature of bacterial oxygen responses can actually be determined by growing the bacteria in culture tubes, which you can see here. And these are filled with a solid culture medium or a special medium like thioglycolate broth, which contains a reducing agent to lower the oxygen levels. And each of the green dots that you can see within these culture tubes represents a colony of bacteria, for example. So if we look at the tube to the left, which contains an obligate aerobe, you can see the bacterial colonies tend to grow near the surface of the culture, where the greater oxygen concentration is present. Whereas if you look at the fourth tube, the strict anaerobe, you can see most of the growth is occurring towards the bottom of the tube, where there is less oxygen. So what's the basis of these different oxygen sensitivities? These different relationships with oxygen are due to several factors, including the inactivation of proteins and the effect of toxic oxygen derivatives. Enzymes, for example, can be inactivated when sensitive groups like sulfhydryls are oxidized. So let's think about oxygen. Oxygen accepts electrons and it's readily reduced because its two outer orbital electrons are unpaired. Proteins called flavoproteins, which function in electron transport, and several other cell constituents and other processes, can actually promote the reduction of oxygen. And the result is usually some combination of the toxic products listed on this slide: superoxide radical, hydrogen peroxide, and hydroxyl radical. Collectively, these toxic oxygen products are referred to as reactive oxygen species, abbreviated to ROS. These products of oxygen reduction are extremely toxic because they oxidize and rapidly destroy cellular components. So a microorganism must be able to protect itself against these toxic oxygen products, or it will actually die. Many microorganisms possess enzymes that afford protection against these reactive oxygen species. So for example, the obligate aerobes and facultative anaerobes usually contain enzymes such as superoxide dismutase and catalase, which both catalyze the destruction of some of these reactive oxygen species. Another important enzyme which can do the same thing is the peroxidase. Aerotolerant microorganisms, on the other hand, may lack catalase but almost always have the superoxide dismutase enzyme. All the strict anaerobes lack both enzymes or have them in very low concentrations and therefore cannot tolerate oxygen at all. Here we have an example of a gas pack anaerobic system, much like the one that we use in the lab to culture anaerobic microorganisms. Hydrogen and carbon dioxide are generated by a gas pack envelope, which you can see there. The gas pack contains a palladium catalyst in the chamber lid, and this catalyzes the formation of water from hydrogen and oxygen, thereby removing oxygen from the sealed chamber. Our last topic in this lecture will look at cell-to-cell communication within microbial populations. For decades, people tended to think of bacterial populations as just collections of individual cells growing and behaving independently. But about 30 years ago, it was discovered that the marine luminescent bacterium called Vibrio fischeri controls its ability to glow by producing a small diffusible substance called an autoinducer. And this autoinducer molecule was later identified as an acyl homoserine lactone, AHL, as described here. And it's been found that many gram-negative organisms produce these AHLs. Now, what they found was that the concentration of the AHL autoinducer molecules within the environment, within the immediate environment, allowed individual microorganisms to assess population density. And this process is now referred to as quorum sensing. And remember, a quorum usually refers to the minimum number of members in a particular organisation needed to conduct business. So how does quorum sensing work? Well, in many of these species of microorganisms, the AHL or other signal molecule is freely diffusible across the plasma membrane. So, well, when cell density is low, the molecule just diffuses out of the cell into the immediate environment. However, when the cell population increases and the AHL molecules accumulate outside the cell, the diffusion gradient becomes reversed, so that the AHL molecule then diffuses from the external environment back into the microbial cells. And because the influx of AHL is cell density dependent, it enables individual microbial cells to assess population density. So this is what's referred to as quorum sensing. Then when AHL reaches a threshold level inside the cell, it actually induces the expression of a number of different genes that regulate different functions depending on the species of microbe. And these functions are normally processes that are only effective if a large number of microbes are present. So for instance, imagine the light produced by one cell of the bioluminescent bacterium. It wouldn't be visible. But cell densities within the light organ of marine fish and squid reach about 10 to the 10 cells per milliliter. So this actually provides the animal with a flashlight effect when all the microbial cells are producing bioluminescence at the same time. And in fact, many of these processes regulated by quorum sensing involve similar host microbe interactions, such as symbiosis and pathogenicity. Here we have photos of the bobtail squid. This is a warm water squid that remains buried in sand during the day and feeds at night. When it's feeding, it uses its light organ, which you can see boxed here on the right, and it's located on its ventral surface. It uses this light organ to provide camouflage by projecting light downward. So the outline of the squid appears as bright as the water's surface to potential predators looking up through the water column. Now, this light organ is colonised by a large number of the bacteria Vibrio fischeri, and they produce these autoinducer molecules which accumulate to a threshold concentration, triggering light production. I'll leave you with some additional examples of bacteria that use AHL signals. So in addition to Vibrio fischeri bioluminescence in the bobtail squid, we have the examples here of the opportunistic pathogens, Burkholderia and Pseudomonas aeruginosa, that use AHLs to regulate the expression of virulence factors. The plant pathogens, Agrobacterium tumefaciens, will not infect a host plant, and Erwinia caritivora will not produce antibiotics without AHL signalling.